So today we're going to talk about hormones and their interaction with endogenous rhythms in our body and how that can impact sleep. All of us have internal clocks built into our biology. These clocks can track things over the course of a day, a month, or a year, or some other period of time. All vertebrate animals have these clocks built into our cells. You can isolate the cell and still see the clock continue its period that is programmed into it. If it's even removed from the system, even bacteria have, some clock-like patterns in their cells. This rhythmic nature makes sense. If you think about it, most animals are also diurnal or nocturnal. Things like the sun and these natural periods of time in nature can greatly shape our biology through the process of phylogeny or the greater impact of environmental and external events on the organism and how that sort of shapes the expression of certain behaviors even from an evolutionary or adaptive perspective. So being able to track and time these patterns and having these sort of built in is, highly advantageous and therefore was selected for. So you can actually measure and see cyclic patterns of release of hormones. Again, some of these would occur over the course of a month, for example, with the menstrual cycle, or over the course of a day, like you can see here, following patterns of luteinizing hormone, in this little rodent here. It would be very common that you would see fluctuations like this in various hormones over periods of days, months, or years. And this is one reason I don't think that we're going to really have a great grasp on the effects that hormones have on behavior until we can measure or monitor these things in near real time, through implantable tattoos or something like this in the future. A single blood draw, for example, in this chart at one point in time might be very problematic if you were to draw conclusions about how luteinizing hormone is associated with a particular behavior, especially if you don't account for the cyclic nature of the hormones being released. So what I'm really talking about today are circadian rhythms primarily. Circa would be around about, and dies from circadian would be from day. These would be corresponding to about a 24-hour period. Most of us actually run on about a 24.5-hour period, but it's very close to being 24 hours. These circadian rhythms are physiological and behavioral characteristics that follow a daily or circadian pattern. These are typically measured in the lab in rodents using running wheels, and you can see very clear, distinct patterns where the animal will run during the active cycle, which would be the dark cycle for them. They're being nocturnal animals, and then they will stop running during the light phase. There are various things that you can do, manipulations to change that pattern of the activity. But it's not that there are other rhythms as well that you should be aware of. There are ultradians, which are cycles that are shorter than a day. They can be as small as the few milliseconds to a 90-minute sleep cycle, like what you might see during REM versus non-REM sleep. REM typically lasts only about 90 minutes in a healthy young adult. Hunger is another example of a ultradian rhythm, or it can be. Infradian would be cycles that last longer than a day. The menstrual cycle would be a great example, or the estrous cycle, hibernation in animals, and bird migration. Of course, we've already gone over circadian patterns. One note here is that various hormones will show patterns over the course of the day. One to note is that testosterone levels typically peak early in the morning around 6 a.m. and then decline to a low around 6 p.m. or 8 p.m. So, there is might be something to this notion of hitting the gym in the morning. So there's also circannual cycles occurring yearly or annually. This would involve hibernation, forms of migration, and some reproductive patterns in various animals that follow particularly an annual mating process. Here's some data from that look a lot like running wheel data here to the left, where you can see sort of periods where the animal would be active or inactive during periods of the lights being on and off, that you can see at the very top on a bar there, where it would be the white would be the light on. This bird, not being nocturnal, would then be moving a lot more. But you can see that during a seasonal pattern here, even if you remove the bird from nature and leave them in a vacuum, constant temperature, constant humidity uh, lab, that you would then see the animal begin to exhibit sort of migratory type behavior, where you're seeing them moving more even in the inactive period. Even when the lights are off, they are exhibiting much more activity. So the idea here is that there's just naturally ingrained here to go and seek out a new place to be at this time of the year. That's just built into them through years of evolution, through the process known as phylogenesis, where even the behavior of the animal can be sort of shaped over time through evolution. It's important to note that we have internal and external controls over these biological rhythms, though. I mean, there are some that are endogenous or built in. For example, like what we just saw in the warbler. You also have different fluctuations in your own body, including even changes in temperature over the course of the day, that are just sort of built in. But there are ways for external triggers to modify these internal cycles to, for example, phase advance you, um, being flashed with bright blue light in the middle of the night, for example, would trigger, some biological mechanisms in you that might result in you not only waking but having a little trouble falling back to sleep, and you could even see greater disturbances later throughout the day. So Zeitgeber is the term that we use for these stimuli, like the bright blue light, the blast of blue light that would be giving you time. In this case, we typically think about the sun, but there are various things that could be Zeitgebers or set, sort of the alter your clock. There are clear environmental cues like sunlight, food, noise, or social interaction. Zeitgebers help to reset the biological clock around a 24-hour day. The process of synchronization of endogenous biological rhythms with a periodic cue in the environment is called entrainment. So, if we were to get on an airplane and fly 14 hours in any direction, especially if we're going left or right on the map, we would probably have to result in go through a period of time where we are entrained to the new light environment, our new Zeitgeber. And during that period where we'd phase advance probably 30 minutes to an hour a day, we might be quite cranky. Interestingly, our internal rhythm is about 24 and a half hours. So if we were placed in a room where it was all dark, you would see us just slightly phase advance a little bit each day. So again, there are different periods. The circadian, which would be tied to the revolution of the Earth. There's also circadial patterns, which would be associated with the tide. Circannular to lunar phases of the moon, and circannual, which would be associated with seasons of the year. These are just common examples in nature which have had really a profound impact on shaping our biology through phylogenesis. Here's just a quick overview of how some of your physiological properties are shaped by these circadian rhythms. particularly when you are younger. I mean, the growth hormone effect sort of becomes less pronounced when you're older, of course, but especially when you're still developing, much more growth hormone is released when you are asleep, uh, which one very good reason that children tend to sleep a little longer than adults. We know that there's one master clock that controls many peripheral clocks throughout our system. So, you might be able to remove, cells, in a peripheral part of your body and see still then maintain a secular or cyclical pattern in vitro or in, a lab, completely removed from our brain even. However, we know that there is a sort of a master clock within the mammalian brain that can control or influence all of the peripheral clocks. And there is also a mechanism for environmental cues like light to entrain it. So they do this by these photoreceptors within the eye being able to detect light, particularly blue light. One reason that blue light is added to lights in the hospital to, essentially keep us awake, even when we otherwise would not be. But these photoreceptors, when triggered with blue light, lead to the activation of action potentials that are transmitted down the retinohypothalamic tract to lead to the release of glutamate into a brain region called the suprachiasmatic nucleus, which contains the molecular machinery that makes up your master biological clock. This is, of course, like I said, the suprachiasmatic nucleus, and there's a lot of evidence to support that it is sort of a master clock. First, it is independent. The suprachiasmatic nucleus can very clearly remain rhythmic in nature in cultures. We've also, and this is really, I think, telling to me, if you remove the suprachiasmatic nucleus from animal A and then transmit it into animal B adopts a general circadian pattern of animal A, and all of their sort of peripheral patterns will sort of snap into line as well. The suprachiasmatic nucleus is also a self-sustaining oscillator that instills rhythmicity in other brain regions through neural connections and can receive input from the photoreceptors through this retinohypothalamic tract, which releases glutamate into it, which is just one mechanism through how it is entrained. The suprachiasmatic nucleus is a small region of the brain found directly above the optic chiasm. It's responsible for controlling our circadian rhythms, and really, there are only around 20,000 neurons here that are part of this master clock. In general, light can entrain the suprachiasmatic nucleus by activating the photoreceptors that would transmit electrical signals through the retinohypothalamic tract to result in chemical release of glutamate into it to phase advance the molecular machinery within it. So what is the molecular machinery that makes it tick? These are, the sort of the gears of the automatic watch. Um, Basically, you just have sort of a teeter-totter between two sets of proteins that are produced or translated by the activation of genes. And these genes, for the most part, are suppressed when the other proteins, the competitor proteins, are present. But they begin to decay over about 12 hours, and then the genes kick in and start translating the other set of proteins that are sort of on the other side of the seesaw. And they just keep going back and forth every 12 hours in a self-regulated feedback loop. The first two proteins involved are clock and BMAL1, which dimerize or form together and enter the nucleus of the cell. These will then decay over 12 hours. While they decay, you have genes turned on called period encrypted chrome that would be suppressed in the presence of clock and BMAL. But period in cryptochrome, as clock and BMAL begin to decay over around 12 hours, begin to synthesize their protein products, period in cryptochrome, which bind together with another protein called tau outside of the nucleus of the cell. And this protein complex will also stay together for around 12 hours until it decays. And then clock and BMAL1 start forming again, back and forth, back and forth. Here's just a figure illustrating this. So clock and BMAL1 bind together, enter the nucleus. When they are together, they over time would turn on the period encrypted chrome genes. The period encrypted chrome gene protein products would then be translated, leave the nucleus, and form a complex with tau. The period cryptochrome tau complex then usually suppresses the translation of clock and BMAL1 complex, so you would not see clock and BMAL1, but over about a 12-hour period in cryptochrome breakdown, resulting in the continued translation of clock and BMAL1, and the cycle repeats itself. So this is the 24-hour feedback mechanism. The period cryptochrome complex suppresses the action of the clock BMAL1 complex, resulting in less activity of the period and cryptochrome. With less period and cryptochrome produced in the degradation of the three, you see this decline over time, which basically takes the brake off clock and BMAL1 production. The entire cycle lasts about 24 hours, with clock and BMAL1 dominating for the first half and period and cryptochrome dominating for the second. We know that the suprachiasmatic nucleus acts on other clocks that can sort of snap them into line. There are surely some that continue operating on their own. For example, you really have a tick-tock mechanism in your medulla that keeps your diaphragm contracting every few seconds. You don't want that to become under control of the suprachiasmatic nucleus, for example. But there are many peripheral clocks and really central clocks as well, clocks throughout the body that can be impacted by the suprachiasmatic nucleus output. So in general, we know that we have this master clock, the pacemaker, and the suprachiasmatic nucleus that leads to altering patterns of chemical release and function and physiological function in a variety of body regions and organ systems. And this would lead to many different changes in behavior. And of course, this can be entrained by the environment, which again is just sort of the output. I mean, this is sort of my general view of how I see everything. Of course, you can have environmental, cultural influence on the sort of the central component of the system, the brain, with the endocrine system, of course, playing a very important role here as well, very connected to the brain, tied together really at the level of the pituitary and hypothalamus. But then this would give rise to changes in our behavior, which is essentially where it originates. But we know that the suprachiasmatic nucleus has at least two major output pathways. One that goes to the preoptic nucleus of the hypothalamus, which is involved in many different behaviors. One that's well established is that this would alter ovulation. The second pathway leads to the paraventricular nucleus in the hypothalamus, which is very important in integrating neuroendocrine and autonomic functions, like those involving your heartbeat, for example. And then it also can lead to changes in the pineal gland, which produces melatonin. We're going to go over that quite a bit here in a minute. Vasopressin and oxytocin and orexin and hypocretin are also known to be very important in modulating these. And I will point out oxytocin and hypocretin are two distinct chemicals. So we know that through that latter pathway, we can alter the function of the pineal gland by changing or entraining the suprachiasmatic nucleus. Melatonin is a very ancient chemical that we think subserves many functions in many different animals. But in humans, we know that it can change our body temperature rhythms. It can facilitate sleep, and it can control photoperiodic responses, or, for example, our responses to changes in light, which is one reason that melatonin is also often recommended for jet lag. Neural connections from the suprachiasmatic nucleus to the anterior hypothalamus initiate many different hormonal events as well. For example, you can see activation of the adrenal gland might ultimately change levels of glucocorticoids, which would then go on to produce very many different physiological functions. All of these as well can be entrained, really going back to the level of the blue light. Going back to melatonin here, this is a hormone secreted by the pineal gland, and it contains light-sensitive cells uh, 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 that really have a connection from the eyes. So right, I mean, essentially there is sensors, receptors in the eye that would also lead to axonal input into the pineal gland as well. But we also know that it's under control of the suprachiasmatic nucleus. The pineal gland converts serotonin into melatonin. So you would have to have enough tryptophan or serotonin to be present to produce melatonin. And this is one reason why people talk about getting sleepy so much on Thanksgiving. The idea here is that theoretically, if you were deficit in tryptophan, it would make you more sleepy. The reality is most of us are probably not deficit in tryptophan if we are maintaining a healthy diet. But melatonin is, again, very ubiquitous in nature. It is one of the very ancient biological signaling mechanisms that you see conserved or used in many different mechanisms in many different species. It also seems to have antioxidant properties, where it would counter free radicals and inflammation. It's also under regulation and is prone to entrainment. It can influence many different things in turn, including your blood pressure, bone density, and regulation of bicarbonate secretion in the GI tract. I think probably one of the more common prescriptions in the country is still for, gastric reflux. Melatonin could be associated with that a little as well. One reason that especially if you're sleeping poorly, you could have difficulties with the reflux. Melatonin is primarily secreted by the pineal gland, of course, and it involves or is synthesized from serotonin. Uh, Interestingly, exogenous administration can have many different effects. Some more, based in strong science and others. There's a lot of speculation out there involving melatonin. However, it has certainly, been, it does definitely influence many different processes, including circadian mechanisms. You can see a clear 24-hour cycle of melatonin production. Melatonin peaks around the early morning, around 3 a.m., and then it begins to decay around the time that you would wake up. And then it begins to rise again around bedtime. Interestingly, melatonin also declines through age. It would peak around the age of 8 to 12. And then in old age, when it really becomes very difficult to sleep, I will add, melatonin essentially goes away. So melatonin could be playing a much more important developmental role in really neurodevelopment even than we give it credit for. Um, we also know that in general, circadian patterns are very impactful on human health, not just melatonin, but I mean, in general, these circadian patterns are very important. For example, most heart attacks and strokes occur between 6 a.m. and noon. These are periods of time where, due to neuroendocrine changes, this is when blood pressure would rise. This is when platelets become stickier and more likely to form clots. And the mechanisms that break down the clots is least active, all due to changes in hormones. In addition, asthma attacks also show a circadian pattern and are most likely to occur at night. Here, the idea is that levels of epinephrine, which causes the air tubules to dilate, and cortisol, which suppresses the immune system, are both low. Another good example is the hormone leptin decreases appetite and ghrelin increases appetite. These can change at different periods of time, but certainly can influence sleep as well. If you are hungry due to changes in ghrelin, which is typically released from the gut as an allosteric process that would be received by the brain, and then go on to impact the suprachiasmatic nucleus or your sleep mechanisms, which I'll get into a little bit here at the end. But yeah, I'll also point out that sleep-deprived humans have low leptin levels and high ghrelin levels, giving them a heartier appetite than usual, and also it's sort of hard to sleep when your ghrelin levels are high. Another fascinating finding was what happened when we were able to generate mice that lack the circadian clock or the molecular mechanisms. They're the genes that are involved in the clock within the suprachiasmatic nucleus. These mice gain a lot of weight. I've worked with clock animals. They are, 3 times as wide as a typical animal. They develop high cholesterol. Their blood sugar and triglycerides are high. They have low insulin and bloated fat cells. These are some, very altered mice. And I think it really just reinforced how important these circadian patterns are in influencing our physiology and our behavior. Here's another important thing to keep in mind. As I've already pointed out, sleep begins to become less prevalent over age. Uh, this is probably at least somewhat associated with things like melatonin declining as you age. However, it's also, I think, important to note that there is a sizable sex difference here, where female humans tend to report greater incidence of insomnia than males, really throughout the aging process, which would likely implicate some effective sex hormones on sleep as well. Uh, Androgen and estrogen systems form distinct connections with the suprachiasmatic nucleus. We're not going to go into the specifics, but just know that there are some distinct feedback loops and these sorts of things between the androgen and estrogen systems. We also know that prolactin also acts on the suprachiasmatic nucleus to influence its output. Prolactin levels also have a circadian pattern with peak levels occurring during the night and being lowest in the mid-afternoon. Recall that prolactin also triggers the milk letdown response, and this is one reason why milk supply would be highest in the early morning, which is, this is, it's something that some people call sort of a cruel irony, but it's just all linked to circadian patterns and hormones producing this physiological response. Prolactin levels are also higher during the luteal phase of the menstrual cycle than during the follicular phase. And, prolactin in general could be playing some role in here. So here in a nutshell is the neural circuitry of sleep. I'm not trying to overwhelm you with this last one here, but this is, I guess, my view of the neural circuitry of sleep. So we primarily covered here what you see in the middle, in this middle oval, the circadian patterns in today's lecture, where we talked about cellular clocks and light signals and how that would feed into different regions of the hypothalamus, including the ventrolateral preoptic area up there above. I should also have a line going to the VLPO in addition to the LH. But we talked about how the retinohypothalamic tract and prolactin can both influence the effects of these sort of circadian clocks on the lateral hypothalamus, which would then go on to influence the pituitary and hormonal release. We also talked a little bit about allosteric signals. One of these would be like the hunger in the gut stimulating release of ghrelin, which is going to then act on neurons within the lateral hypothalamus to drive you toward feeding, if not other Fs, in sort of a generalized state of arousal. But really, that is what the lateral hypothalamus is directly tied to, is the arousal network, also known as the reticular activating system in sort of older texts. But the arousal network is composed of neurons that form distinct clusters of acetylcholine, norepinephrine, serotonin, dopamine, and histamine in different regions of the brain. Really, with all of those but histamine, we've now shown with optogenetics that you can activate these specific neurons to arouse the animal and awake them, or really produce the opposite of sleep. Anyway, and one other very important hormone that seems to be right in the middle of all of this is something known as orexin or hypocretin, which is what we're going to end on here. Orexin is a neuropeptide hormone that's produced in the lateral hypothalamus that really innervates all components of the ascending reticular activating system. I myself have studied how orexin alters dopamine neurons and dopamine-associated behaviors like feeding and drug-taking behavior. It does. It's a very clear effect, I would say. But it also has clear effects on all of these other arousal network systems as well. And therefore, seeing how it is a neuropeptide hormone, I wanted to spend a minute talking about it as well. So we now know that there are very few orexin neurons. Orexin neurons are also called hypocretin neurons, I would like to point out. Orexin and hypocretin were discovered, the neurons, by two distinct labs at the same time that named them different things. Orexin mainly indicates that it is a drug or a chemical that would make you go eat, go feed. Of course, this is one function associated with the lateral hypothalamus. There are others. But in general, it is arousing. It tends to wake you up and do all, generally would also make you go seek out food, in addition to other things as well. But orexin and hypocretin, we know, accumulates over periods of waking. Antagonists of the orexin hypocretin system promote both non-REM and REM sleep. We also know that optogenetic activation of orexin hypocretin neuron promotes and sustains wakefulness. Here you can see a mouse. It's about to receive optogenetic stimulation. They're about to turn on their optogenetic neuron or their orexin neurons like you would a light switch. In the background there, you can see an EEG on top and an EMG on the bottom. You're about to see a change in slow wave sleep, like probably something in the delta range to beta waves here. Sort of, you can see they got shorter there, and then below that, what you see the EMG measuring the motor activity. So they essentially just woke up that animal by turning on their hypocretin neurons. We also know a lot about the orexin hypocretin system through work on dogs, particularly Dobermans, who have a genetic alteration in their orexin hypocretin system that causes narcolepsy. Many of you have probably seen videos like this, or maybe in person, an excited dog, particularly if you give them a hungry dog access to food, they might get very excited. And if they have this genetic mutation associated with the orexin hypocretin system, they will fall right to sleep for a little bit. In humans, we know that drugs that act on the dopaminergic part of the arousal system, which orexin and hypocretin also work on, like methylphenidate or modafinil, these sorts of stimulant drugs can counteract that narcolepsy. But yeah, I mean, again, here, think of that as being sort of the arousing component of the dopamine system, which would be further downstream to orexin, which also shows the circadian pattern. But there, orexin is also tightly interlinked between the suprachiasmatic nucleus and those arousal populations or of the reticular activating system that all really feed in together and interact with the hypothalamus to result in the regulation of hormone release to alter physiological clocks throughout our body.